The overall goal of this procedure is to assay RNA editing using functional editosomes extracted from Trypanosoma brucei for rapid identification of the editosomes inhibitors. This is accomplished by first isolating mitochondria and the target editosomes from T. brucei. The second step is to prepare the synthetic RNA editing reporter substrate. Next, compounds are subjected to binding to purified editosomes along with the RNA editing reporter. In the presence of compounds that block the editosome's function, the RNA reporter will not undergo editing and will remain inactive. The final step is to add the FRET substrate and monitor for the RNA editing reporter's ability to selectively and specifically cleave this substrate. In the case of an inhibitory compound, RNA editing is blocked and a reduced or no fluorescence signal will be detected. Ultimately, a real-time PCR machine is used to monitor the fluorescence emitted from the FRET substrate as a function of time.
The main advantage of this technique over conventional in vitro editing assays is that it's a simple and rapid mix-and-measure fluorescent-based reporter assay that can be used for high-throughput screening of chemical compounds against RNA editing.
To begin, prepare growth medium for T. brucei procyclic form cells as described in the accompanying text protocol. Next, grow 300 milliliters of T. brucei 1.7a wild-type procyclic form cells at 28 degrees Celsius while shaking them at 70 rpms for 48 hours. Once the cells have reached a density